we're going to review the organelles, cell structure. This is just going to be a quick review, and there's obviously more detail in your notes, so you want to make sure you know these details. But this is a way I find helpful to study for cells and organelles. So let's start off with our plasma or cell membrane. And this is going to be the structure that regulates things moving in, regulates things moving out.
Yep. It's kind of walls of the...
Yes, kind of the castle wall that keeps things either in or out. In the middle, we have our nucleus. This is what I think of as our command center. This is where we're going to find our DNA. In the center of the nucleus, we often find our nucleolus. And this is going to make our ribosomes.
Yep, because there is the rough ER and a smooth ER in there.
Yep, that's coming next.
Okay.
So around the nucleus, the nucleus is surrounded by a nuclear envelope. The nuclear envelope acts as a membrane around the nucleus. Both the plasma and cell membrane and the nuclear envelope, both of them are bilayers, meaning there's two layers.
2 layers, yep.
All right. Off of the nucleus, extensions of that, we're going to have our endoplasmic reticulum. Collectively, I think of the endoplasmic reticulum as kind of multitaskers. They do a lot of different things. But we do have two versions. We have our rough and our smooth. Our rough, if you look on the pictures of it, you're going to see these teeny little dots. Those are going to be our ribosomes. Our ribosomes are our builders. They're going to build our proteins.
Okay, so they build proteins.
Yes, and they're found in the rough ER. So visually and functionally, that's one way we separate out the rough ER is this is where proteins are going to be made. And they're going to be made by the ribosomes that are found here. So that's one of the big distinctions. Yeah. Our smooth ER is really important in detoxifying the cell. So it's in a, that's one of its roles. Cleans up the cell.
Toxic stuff. Okay.
Yep, so it helps cleans up the cell. We also have, we have our mitochondria.
When I was back in high school, we used to call it flip-flop.
It does. It looks like the bottom of a flip-flop.
Yeah.
It's kind of bean-shaped.
Yeah.
It has these little cristae, these little shells in here. You can also find some ribosomes in here as well, and ribosomes can also float out in the cytosol here. But mitochondria is what I commonly think of as our powerhouse.
Oh, you know what I was confusing? Nucleolus and mitochondria. I was confusing which one was the powerhouse.
Yep. The mitochondria is the powerhouse. This is going to synthesize ATP, or put together the ATP molecule.
So makes ATP, synthesizes it, and that's all
Yep.
whatever energy needs to be
Yep,
made.
so
Okay.
it provides the energy for the cell. It doesn't necessarily use the ATP, but it makes it.
It makes it.
I think it's kind of like a coal factory, where the factory makes the coal, doesn't necessarily use it. It ships it out to where it needs to go. That's our mitochondria. Also with mitochondria, it has its own little DNA, a little ring structure of DNA. That's what gets passed on from mother to child.
That's RNA?
Mitochondrial DNA.
DNA, okay. Yep. Oh, yeah.
So it's just a little tidbit about mitochondria.
Okay.
Also in our cell here, we have our Golgi complex and vesicles. I think of the Golgi complex as UPS, packing and shipping.
So they distribute things.
Yep, they distribute things. They top off and package those proteins that were made by the ribosomes. So in my mind, I'm thinking of the Golgi complex as UPS, packing and shipping. The vesicles are the little trucks, move things about. Let's see here. Our lysosomes, these I think of as our sanitation crew. They go around and they break down particles or decaying organelles. In the liver, they help break down our complex carbohydrates into glucose. So they're breaking down. They have digestive enzymes in them. So we're breaking down, helping keeping the cell healthy and clean and functional.
So we can say like our Lysol cleaning stuff is.
Yep, Lysol cleaning.
Yeah.
Yep, with our lysosomes. Yep.
Lysosomes, yeah.
Exactly. Also, we have our centrioles.
Oh, yeah.
Which are nice long protein tubes.
Those are cute.
Yeah, they look like little stars when you look down them.
Mm-hmm.
These are very useful in pulling the cell apart, so they're going to help in cell division. So we have our centrioles. And it all sits in our cytosol, the fluid that's inside the cell. I think of the cell as Jello salad. The whole cell is this whole salad. All the fruits and nuts you put in there are the organelles, and the Jello is the cytosol.
Yeah.
What it all floats in. Obviously, there's more detail for each individual organelle, but if you can sit and draw this out, both with lab and with lecture, when you think about, well, what does the mitochondria look like? How does it look different than the picture of the smooth ER? And the smooth ER looks like water squished under glass. So if you can visually think, how do these look different from each other? What are their functions? And talk through it while you draw it, you're making this a very active process.
Okay.
So this is a good place to start studying.
That makes sense, yeah. I mean, draw what's missing. Well, this one is missing. This one does that, and it's there, and you know.
Exactly.
Okay.
Any questions?